Okay, so let's talk gluconeogenesis and buffer systems. Before we do that, I want to say or talk about amphibolic intermediates and anaplerotic reactions. Amphibolic intermediates are intermediates that exist in, for example, the citric acid cycle that we can use for other pathways. And this will be important today for gluconeogenesis. And if we use one of these, let's say, for example, we will talk today about using oxaloacetate. If we use oxaloacetate from the citric acid cycle, then we have to do another reaction so that produces oxaloacetate, so that oxaloacetate can exist again for the citric acid cycle. And such reactions we call anaplerotic reactions. So for example, if we use oxaloacetate, then we will use pyruvate plus bicarbonate ions so that we can produce oxaloacetate, for example. And such anaplerotic reactions, it is by doing a carboxylation of 3 carbon intermediates into 4 carbon intermediates. That's what I've written here. Okay, so gluconeogenesis, it is the synthesis of glucose from non-carbohydrate substrates. And why? So what's the main purpose of gluconeogenesis? Why do we have it? And it is mainly for the regulation of blood glucose levels, mainly for this reason. So that's why we have gluconeogenesis. For example, if you've been fasting or after intense exercise, then the blood glucose levels will drop, and therefore we need to maintain these blood glucose levels, so gluconeogenesis will occur to increase or keep the blood glucose levels stable. And I think blood glucose levels are around 5 millimoles per, wait a second, it's 5 millimoles per liter. So glucose is the main energy source for the central nervous system, peripheral nervous system, so brain and neurons, and also erythrocytes. Erythrocytes lack mitochondria, therefore they will use glycolysis, only glycolysis, therefore they need a lot of glucose. The testes, renal medulla, and embryonic tissues will also be highly dependent on the glucose molecule. So, where does gluconeogenesis take place? It takes place in the kidney, enterocytes, but primarily in the liver, and it's in the cytosol of the hepatocytes. So it's in the cytosol of liver cells. That's where gluconeogenesis will occur. Okay, so this is basically what happens. The liver here we have the liver. Here we have liver cells. Inside of these liver cells, so inside their cytoplasm, there will be gluconeogenesis, so we will create new glucose molecules. And what happens is that when we have created these glucose molecules, they will diffuse out, and then they will go to the blood, and then, they will go to the blood, and we will regulate the blood glucose levels. Yes. And it's also very important to remember that it's when we fast and after intense exercise that we will do gluconeogenesis. So gluconeogenesis is the opposite of glycolysis. So here we have the glycolysis pathway. We start from glucose and then we will go to pyruvate. Gluconeogenesis goes from pyruvate back to glucose. But the thing about gluconeogenesis is that we don't have to start from pyruvate. We can start from lactate, and lactate can go back to pyruvate, and then from pyruvate back to glucose. Or we can start from glycerols. It's specifically glycerols and not fatty acids. So and I think we will talk about this in next semester, is like fatty acid breakdown will give us glycerol. And these glycerols will give us, wait, let me just, okay, no, never mind. Okay, glycerols from the fatty acids can go into the gluconeogenesis pathway, and also amino acids. So amino acids can also be converted into intermediates of glycolysis, and they can be used for gluconeogenesis. Also, glucogenic amino acids can also be converted into citric acid cycle intermediates, and then the cycle can go back to gluconeogenesis. Okay. If you remember from glycolysis, there are 3 steps that are irreversible. It was step number 1 of glycolysis, step number 3, and step number 10. These steps are irreversible. So when we say that gluconeogenesis is the opposite of glycolysis, this means that these 3 steps we have to overcome, even though in glycolysis you cannot bypass them in glycolysis. But in order to do gluconeogenesis, you would have to bypass these 3 reactions, and we can do that by specific enzymes and by specific enzymes and by vitamins that will aid, and cofactors that will aid in that. And I think this is important for the colloquium. We have to know these enzymes that go back to that help gluconeogenesis bypass these 3 reactions. And gluconeogenesis doesn't make ATP also. That's important to know. But we will talk about calculations also. So these are the 3 steps that have to be bypassed. So I will just go quickly over. So here we have glycolysis. Here is step number 1. Where is step number 3? Here is step number 3. And step number 10 right here. So these 3 reactions are the irreversible ones. And obviously the first step, so gluconeogenesis will start from here to glucose. So the first step we would have to overcome is actually step number 10 of glycolysis. And step number 10 of glycolysis, it was just like PEP or phosphophenols pyruvate together with pyruvate kinase, which created pyruvate. And in that we gained 2 ATPs. But in gluconeogenesis, we will start from pyruvate, and we want to get to phosphophenols pyruvate, just like I written here. The thing is that in order to do that, we have to do 2 reactions. So here, from glycolysis, we only had 1 reaction that went to pyruvate, but in order to go from pyruvate to phosphophenol pyruvate, we need to do 2 reactions. The first one is to go from pyruvate to oxaloacetate. And we do that by help of pyruvate carboxylase, and then also biotin, which is B7 vitamin, and also ATP. So this is also really important to remember for the colloquium. Then we will go from oxaloacetate to phosphophenol pyruvate, and we will do that by phosphophenol pyruvate carboxykinase and 1 GTP. And also it's important to remember that we will lose 1 carbon here, so we will lose it in the form of carbon dioxide. Yes. So here's another thing I would like to mention about this step. So we can start from pyruvate. Pyruvate can travel into the matrix of the mitochondria, and then we can convert it into oxaloacetate, just like we did in this in this step. But the thing is that oxaloacetate cannot go out of the cell. It cannot go out. It doesn't have any transporters. So therefore, oxaloacetate will be converted to malate, and then malate malate in the cytosol will be converted into oxaloacetate. Then we can do this. Then we can do step B into phosphophenylpyruvate. And the fact that this reaction can go like this is because NADH plus H plus in the matrix of the mitochondria is in high concentration, but in the cytosol it is in low concentration. So in order to equalize this, then the reaction will go actually this way, and then that's really good because then we can get to phosphophenylpyruvate. And if we start with lactate, we can also start from lactate, and then we can go to pyruvate, and then pyruvate will be transported in, and then oxaloacetate, and then directly we will get PEP. And PEP can actually be transported through transporters out of the cell. Okay. The second step that we have to bypass is step number 3 of glycolysis, but it's when we go from fructose 1,6-bisphosphate to fructose 6-phosphate. So what happens is we will do that with help of fructose 1,6-bisphosphatase 1. I don't know why I didn't highlight it red, but yeah, 1. And this is oppositely regulated by phosphofructokinase cleavage by water. And there's no ATP gain here. So this is important for the calculations, and I want you to remember that, is that when we go from fructose 1,6-bisphosphate to fructose 6-phosphate, we will have no ATP gain or loss, none of that, because this enzyme only takes away a phosphate from the fructose. It doesn't give the phosphate, it doesn't do any substrate level phosphorylation, is what I mean. And in that case, we will get no ATP. And also, it doesn't use ATP to do that. So no ATP gain or loss, and the same thing goes for this reaction number 1. So for reaction number 1, we have to dephosphorylate glucose 6-phosphate, and we do that by glucose 6-phosphatase. So, and then after that, it can be transported out of the liver, and then we have a glucose molecule, and that glucose molecules can go into the blood, and then we do the maintenance of the blood glucose levels. The Cori cycle is recycling of lactate into glucose. So there's a rapid movement. So there will be glycogen breakdown, and we will produce lactate. Lactate will diffuse into the blood and then into the liver. And then lactate here will undergo gluconeogenesis, and it will become glucose, and then glucose will go into the blood, and it could then also enter the muscle, or it could just stay in the blood. Yeah, that's the Cori cycle. So I hope I just made it clear that we had the different substrates. So pyruvate, the different substrates for gluconeogenesis is lactate, amino acids, glycerols, and also pyruvate. And that's what I'm going through. So the Cori cycle will use lactate. And then we have a glycerol that can also be a substrate for gluconeogenesis. And what happens here, what's important to remember here is also the glycerol kinase enzyme. So glycerol, here we have a glycerol. Then the glycerol together with glycerol kinase will become glycerol 3-phosphate. Then glycerol 3-phosphate together with this enzyme glycerol 3-phosphate dehydrogenase will give us dihydroxyacetone phosphate, and that through triose phosphate isomerase will give us glyceraldehyde 3-phosphate. And this is an intermediate of glycolysis. So glycerol basically can go to this molecule right here and then enter the glycolysis pathway, but then it wouldn't continue to do glycolysis, but it would do the opposite. It would do gluconeogenesis, and that's how we can make a glucose from glycerol. Okay, now we can do calculations, and you don't have to read this right now, but I just want to mention, I will go through every single thing, just that in which conditions, daily situations, each of these substrates are going to be more primary source for synthesis of glucose. So lactate, we will use lactate as a primary source during sport. So that means we will use the Cori cycle. Glycerol will be used between meals, and pyruvate and alanine during starvation or protein-rich diet. Okay, and I will talk about this now when we do the calculations. Okay, so calculations of gluconeogenesis if the substrate is pyruvate or alanine. I just want to say that if we have alanine, which is an amino acid, if we have it, it can actually be converted into pyruvate by transamination. So just know that. So the calculations will be the same for these two, because alanine can be converted into pyruvate without using ATP or gaining ATP or NADH or whatever. So for us to do these calculations, let me just do this. Okay. So, when we go from pyruvate to oxaloacetate, we will use 2 ATP. And that's in the case of 2-pyruvate. 2-pyruvate equals 1 glucose. So when we do gluconeogenesis and we want 1 glucose and we will start from pyruvate, we will use 2 pyruvate molecules. So 2 pyruvate molecules will be converted into oxaloacetate, and that will use 2 ATP. So minus 2 ATPs here. Then from oxaloacetate to phosphophenylpyruvate, so this step, we will use 2 GTP. So that's again an investment of 2 GTPs. So far minus 4 ATPs, because 1 GTP equals 1 ATP. That's one thing. Then we will go further up in the reaction. And here we can see that from 3-phosphoglycerate to 1,3-bisphosphoglycerate, we will use 2 ATPs right here. So that's minus 2 ATPs. Then from 1,3-bisphosphoglycerate to glyceraldehyde 3-phosphate, we will use 2 NADHs. So that's minus 2 NADH. This equals, in total, minus 7 ATP. So here we have minus 4 ATP, here we have minus 7, that equals, so far, minus 11 ATPs. Then we go further up. And if you remember what I just said, is that the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate will not give us any energy. It will not use or give any energy. It just takes away a phosphate. And the same thing goes for this step. So therefore here it's, I don't know if you can say plus minus 0, but like no ATP gain or loss. Nothing happens. So this means that from 2 pyruvates, we will invest minus 11 ATPs. And this will be the calculations for gluconeogenesis from pyruvate or alanine. It doesn't matter. What else did I want to say? Yeah, here I just written the calculations for the glycolysis. So if you want calculations for glycolysis, just watch the video from that. The second substrate for gluconeogenesis was lactate. And if we used lactate for gluconeogenesis, this is called Cori cycle. So if we want 1 glucose molecules, we will have to use 2 lactate molecules. So when we convert, when we go from lactate, because it's gluconeogenesis, so when we go from lactate to pyruvate, we will use NADH. Okay, so this reaction is going mostly this way. This picture isn't really good for what I want, but just try to reverse it in your mind. But basically, from lactate to pyruvate, you will use I was right. Okay. So from lactate to pyruvate, you will actually gain NADH, because when we go from pyruvate to lactate, we gain an NAD+, and when we go from lactate to pyruvate, we will gain NADH. And if we do this 2 times, because we need 2 lactate molecules, then we will use 2 NADHs, or we will produce 2 NADHs plus 2 NAD, which equals 5 ATP. And then once we get to pyruvate, then the thing will be the exact same. So we will do this, blah, 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 minus 11 ATPs. It will be the same types of calculations. So once we get to pyruvate, everything will be the same, the minus 11. But here we have to remember that we have actually gained 5 ATPs from converting lactate to pyruvate. So this means that minus 11 plus 5 equals plus wait a second. Minus 11 plus 5, it will be minus 6 ATPs. I don't know why I written plus 6. It's minus 6 ATPs. So the net ATP, if we use lactate, will be minus 6. Then calculations of gluconeogenesis if the substrate is glycerol. That means we go from glycerol to glycerol 3-phosphate to dihydroxyacetone phosphate, and then dihydroxyacetone phosphate will be converted to glycerol aldehyde 3-phosphate, which is this molecule right here. So we will go from dihydroxy, DHAP, to GA3P. And then once we get to GA3P, then we have entered the glycolysis pathway. But when we go from glycerol to glycerol 3-phosphate, we will invest 1 ATP, so minus 1. And then we will gain 1 NADH when we go from glycerol 3-phosphate to dihydroxyacetone phosphate. So that is plus 2.5. So 2.5 it's minus 1 ATP plus 2.5 will give us 1.5 ATPs for 1 glycerol, but we will have to use 2 glycerols, so there will be net ATP times 2 equals 3 ATPs for 2 glycerol molecules, so that we can get to 1 glucose molecules. So yeah, and once we've entered glycerol 3-phosphate and we start making, so we will go up to glucose, we will have these plus 3 ATP. Okay, buffer systems in the lungs, kidneys, and the erythrocytes. So the most important thing to know here is the bicarbonate buffer system. And the bicarbonate buffer system basically says that carbon plus water will give us carbonic acid. And this is due to the help of carbonic anhydrase enzyme. And once we get carbonic acid, we get hydrogen plus bicarbonate ions, and these will help in the maintenance of the pH levels. So this buffer keeps the pH at 7.35 to 7.45, and the kidneys are the ones that produce the bicarbonate ions. Also, another thing to remember is that lactate, once it's produced, it actually produces hydrogens together with it. So if we look here at the reaction, pyruvate to lactate will actually give us hydrogen ions right here, and this will increase the acidity. So once we produce lactate, we will increase acidity. And this acidity is neutralized by the bicarbonate buffer system. Okay, where does this carbon dioxide molecule come from? It comes from the tissues due to cellular respiration. And so here, the carbon dioxide will diffuse into the blood system. There, it will react with water, and together with carbonic anhydrase, it will form carbonic acid. Yes. Also, I will talk about the Bohr's effect here, but this is a picture. This is also another slide about it, but I will talk about it here. So, Bohr's effect basically describes the affinity to oxygen, or the affinity of hemoglobin to oxygen. So if we have the higher the carbon dioxide concentration will be, the less affinity hemoglobin is going to have to oxygen. And so that means that it will not let go of the oxygen molecule. So in the lungs, if the pH, the higher the carbon dioxide concentration is, the more acidic the blood will be, and this also means that there will be less affinity to hemoglobin, so the oxygen molecule will not, the hemoglobin will not let go of the oxygen, meaning that the hemoglobin has higher affinity to the oxygen. And yeah, we will talk about the diseases or that are caused due to this. Yes, what else do I want to say? Also, in the lungs, when we breathe out carbon dioxide, this will actually increase the affinity to oxygen, or of the affinity of hemoglobin to oxygen. So we breathe out carbon dioxide, and that leads to a more basic environment, not necessarily basic, but it's less acidic. And this type of environment is good for hemoglobin to bind to oxygen. But please read what I've written here, and hopefully you'll understand. Yeah. And so also this is Bohr's effect. So when pH is low, the affinity to oxygen will decrease. And if pH is high, the affinity to oxygen affinity to hemoglobin will increase, meaning it will decrease, and affinity to hemoglobin will, oxygen affinity to hemoglobin will increase, so the oxygen does not get released when pH is more basic. Yeah. Okay, so the kidney's role in maintenance of pH. So here we have the kidney, blah, blah, blah. So if there is a highly acidic environment, or let's say there's high acidic environment in the blood, what the kidneys will do is that they will take up bicarbonate ions from the urine and transport it into the blood. And when that happens, the bicarbonate ions will react with these, and they will form carbonic acid and will neutralize the blood. So, at the same time, the kidney will take hydrogen ions from here and then transport them to the urine, and in that way they can regulate the pH. And this happens in acidosis, but similarly can happen when there is alkalosis. Okay. So disorders of the acid-base balance. Okay. First and foremost, look at this formula. And by this formula, I mean this one right here. So acidosis means that there is an increase in pH, or a decrease in pH. This means that we have high amount of carbon dioxide, because the higher the amount of carbon dioxide, if we look at this formula right here, if we have high amounts of carbon dioxide, we will increase hydrogen concentration and pH will be more acidic. And so that is what acidosis is. If we look at the formula here, if we have increased carbon dioxide, so if we have a high number of carbon dioxide here, this means that pH will be lower. Similarly, if this one is if carbonic acid decreases, then this will also lead to lower pH. And if bicarbonate ion decreases, that is because of these things. So diabetic ketoacidosis or lactic acidosis, because lactic acidosis, as I told you, will produce these hydrogen ions, and they will react with bicarbonate ions, and this will lead to a decrease in bicarbonate ions, and a decrease in bicarbonate ions, a decrease in this one will lead to a decrease in pH, meaning the environment will be more acidic. So that's one thing, or severe diarrhea, and then you will also lose your bicarbonate ions. Yes. Secondly, an increase in carbon dioxide can be due to chronic obstructive airway disease, or cardiac arrest, or depression of respiratory center, or chest deformities, which basically means that you cannot breathe all of the carbon dioxide out, meaning that the carbon dioxide will stay in your body, and this will lead to lower pH. Alkalosis is, once again, just look at the formula. Alkalosis means that there will be a higher pH, and this can be due to higher amounts of bicarbonate ions. And we will get higher amounts of bicarbonate ions if we vomit. So that will lead to loss of hydrogen ions. If you vomit, then the gastric juices will be, vomited as well, together with the hydrogen ions, but the bicarbonate ions will stay, and this will lead to an increase of them, etc. Secondly, there can also be the increase of or decrease of carbon dioxide. And decrease of carbon dioxide can be due to hyperventilation, which can happen in anxiety or when you have a fever. So when you breathe a lot or you breathe fast, so this can lead to you breathing out a lot of carbon dioxide. And if you breathe out all of your carbon dioxide, or not all of it for sure, but if you breathe out carbon dioxide, so a decrease in carbon dioxide, So if we decrease this one, then this will lead to making more of the bicarbonate ions. And if we have more of that, then this will lead to a higher pH. And I think that's also why, like when people have panic attacks, then they have to breathe into a paper bag, because they will breathe out the carbon dioxide into the bag, and then when they inhale again, they will inhale the carbon dioxide, and this will not lead to alkalosis. And so symptoms of alkalosis and acidosis, we also have to know for the clock, and it's this one right here. So if you have acidosis, then you will have labored and deep breathing, nausea and vomiting, generalized muscle weakness, headache, drowsiness. And then alkalosis is confusion, muscle twitching, lightheadedness. Yes. Okay, but that was it for gluconeogenesis. Thanks.